Good afternoon and welcome to the NINDS Biomarker Q&A webinar. My name is Rakonda Metley and I am the Operations Coordinator. We will start off with a presentation of slides followed by questions and answers. Please submit your questions in the Q&A section featured below and they will be answered at the end of the presentation. This presentation will be recorded and placed on our YouTube page. I will be sending that link along with the presentation slides to everyone that has registered. I will now turn it over to Dr. Taylor Birds.
Thank you, Rakonda, and thanks everyone for joining us today. I want to introduce the biomarker team really quickly. We're in the Division of Translational Research. I'm Carol Taylor Birds. I'm the Program Director for the NINDS Biomarker Program. Dr. Adisa C. Liu is the New Health Program Specialist with our team. You just met Rakonda. And then Ramadra Chandran is the Program Director for the Heal and Pain Biomarker Program. And Mary Ann Pellmanter, some of you may know, she initiated the Biomarker Program some years ago and has now moved on to therapeutic programs, but still is part of our team and a consultant. And then we're really happy to be joined today by Chris Swanson-Fisher, who's in our Division of Neuroscience and is a program officer for the NINDS Biorepository and several of our other resources here at NINDS. And she's going to take a little bit of time to talk about the BioSend repository and how to use that at the end of the presentation. So briefly, we're just going to go over the program goals and terminology first. And then we're going to talk about the specific funding opportunities that are available through the NINDS Biomarker Program right now. And then, as I mentioned, Chris is going to talk about BioSend, and we'll open up for question and answers after that. So NINDS's Biomarker Program is in our Division of Translational Research. And the program is designed to facilitate the validation of biomarkers and biomarker signatures as fit-for-purpose tools for use in clinical trials to accelerate therapeutic development and aid in decision-making for their patient care. And why is this needed? So we know that biomarkers help facilitate the advancement and approval, regulatory approval for therapeutics. And just as an example of this, bioindustry analysis publication that came out last year showing how biomarkers, especially in the phase 2 to phase 3 transition, really help improve the probability of success for getting a therapeutic advance to successful approval. And overall, they're helpful in seeing successful translation of drugs and therapies to the clinic. So what is a biomarker? So a biomarker is a defined characteristic that can be measured as an indicator of normal biological process, pathogenic process, or response to an exposure or intervention, including therapeutic interventions. And this is the definition from the FDA NIH BEST glossary, the biomarker working group. And then for the purpose of the funding opportunities here, we also use the term biomarker signature. And this is a combination of the multiple variables or components to yield a patient-specific indicator that would be used just like a single biomarker. And then we talk about detection methods as well. And this is a catch-all term to describe the method used to measure the biomarker or biomarker signature. So that could be the assay, the imaging protocol, the detection method, and so on, really depending on the modality. Now, I mentioned that biomarker signature is kind of a general term, and other people might refer to this as a biomarker signature, composite biomarker, or multi-component biomarker, or you might have a multimodal component biomarker. And this is a topic that the FDA actually recently just had a workshop on. And I'd encourage you, if you're thinking about a more complicated biomarker signature, to check out the link here and look at some of the videos of the discussions around how the FDA is thinking about these types of biomarkers. So what is the biomarker validation process? So at a really general level, obviously you have initial discovery and development. You're developing your detection method. You're just identifying the biomarker and how it relates to the underlying physiology and how it relates to the clinical outcome or interest or the process of interest. And then it moves on to analytical validation, clinical validation, and can go on to qualification through the FDA or other regulatory body. And of course, the amount of evidence needed as you progress through this validation pathway increases and more is necessary. So a few years ago, the FDA published the FDA Biomarker Qualification Evidentiary Framework Guidance for industry and FDA staff. And one of the points that was made is that often in therapeutic development, the biomarkers that are proposed as part of therapeutic development don't have the adequate data or information that's necessary to support using the biomarker for the proposed context of use within the study design, the trial design. And so part of the rationale and need for the NINDS Biomarker Program is to have funding opportunities that help investigators develop that evidence and data so that you can have tools that are ready for use in clinical trials. Now, analytical validation is a specific term meaning the establishment that the performance characteristics of a test tool or instrument are acceptable in terms of its sensitivity, specificity, accuracy, precision, and other relevant performance characteristics using a specified technical protocol. And essentially, this just means, can I trust the results of the measurement, and what type of validation do I need to do to know that? Clinical validation, on the other hand, is to establish that the test tool or instrument acceptably identifies, measures, or predicts the outcome of interest. Right? So here, rather than do I trust the measurement, are the results meaningful? Can they be interpreted for making decisions at the individual participant or patient level? And then finally, with biomarker qualification, this is the conclusion based on our formal regulatory process that within the stated context of use, the medical product development tool can be relied upon, right? So can the biomarker be reliably incorporated into a clinical trial? So part of the evidence needed and terminology used for our program and for the FDA are the definitions of the biomarker category. And these come again from the biomarker endpoints and other tools resource that was published by the FDA and NIH. And it's important to look at these descriptions and definitions as you're putting together your proposals, because it tells you how the biomarker is going to be used and therefore what kind of evidence is needed to support it for that use case. Now there's 7 general biomarker categories: diagnostic, monitoring, pharmacodynamic response, predictive, prognostic, safety, and susceptibility risk biomarkers. And this resource also talks about surrogate endpoints or reasonably likely surrogate endpoints too. And so I'd really encourage you to take a look at these. There's examples and a little more description in the glossary. Now, context of use is another really important concept, both for the funding opportunities here and in general as you're thinking about validating your biomarkers, because it describes how the biomarker will be used. So it's a concise description describing both the best biomarker category and how the biomarker will be used in the therapeutic development or clinical practice. So some examples of these, excuse me, or you might have a diagnostic or a prognostic biomarker to use to stratify or enrich patient populations so that you're targeting the right therapeutic to the right patient population. You might have a pharmacodynamic response biomarker that's being used to demonstrate therapeutic target engagement in the tissues of interest, and might be used for informing dosing decisions in your phase 1 or phase 2 trials. And then, of course, risk biomarkers to indicate the risk of developing a disease or condition, monitoring biomarkers to monitor disease progression or changes in response to an intervention, and predictive biomarkers to help inform treatment decisions. Is this patient more likely to respond to a particular intervention or not? So again, and I'm going to say this multiple times, right, but the NINDS Biomarker Program is really designed to help studies do rigorous validation for biomarkers for specific context of use. Now at NIH and at NINDS, there are lots of different funding opportunities where biomarker discovery work and development work can be done. And the distinction here with this program is it's a really focused program to help validate biomarkers as tools for use in clinical trials or clinical practice. So these are the funding opportunities that are currently out. We start at the development stage with an R61, R33 funding opportunity. And then we have cooperative agreement program announcements for both analytical validation and clinical validation. We have both a U01 and U44 small business options for those. So we're going to talk about each of these more in a little bit detail, but the development, PAR 22089, is to conduct that proof of concept studies in human samples, data, and to determine if the candidate biomarker is fit for purpose. So it's going to do your initial clinical validation to identify, can this biomarker be used? Can it be measured for a specific use? And then the later stage cooperative agreements are set up so that you can do more rigorous multi-site validation of the detection method, and this needs to be for a defined context of use. And then the clinical validation, finally, where you have multi-site validation in a really large representative patient populations to obtain the data you needed if you're going to do FDA qualification or demonstrate readiness for use in clinical trials. And for all of these program announcements, the next receipt date is June 22nd. So I also want to point out we have a couple of NOCs on the NINDS biomarker funding opportunities. The first one of these is with what Ram Ardachandran is the point of contact for, and these are a special interest for biomarkers to predict or monitor response to a therapeutic or intervention in pain studies within NINDS mission space. And then the second is Kodun Lungu from our Division of Clinical Research has a notice of special interest out for functional neurological disorders. And so for any of these, you're welcome to reach out to them or to myself, and we'll both join a call if you want to discuss an aim page and you're fit for the project and the program. Now, I'm going to talk about PAR 22089, and I want to point out that this is a reissue of PAR 19315. So for those of you that were familiar with that program, I would really encourage you to make sure you read through the application because there are a few things that have changed in the reissue. NINDS and NIA are both signed on to this funding opportunity. So I'm going to be talking about NINDS specific programs here, but if you're interested in applications in the NIA space, I'd encourage you to take a look and reach out to Dr. Liu, who is the NIA contact for this program announcement. And the first receipt date for this reissue is going to be June. Now, PAR 22089 is a phased mechanism. So you can have up to 5 years, and the phases can be split either as 2 to 3 years in phase 1 or 2 to 3 years in phase 2. But again, total years can't exceed 5 for the full duration. And then budgets aren't limited, but if you request more than 500,000 direct costs in any given year, then you need to reach out to us at least 6 weeks in advance in order to get prior approval from leadership and get through that process to get permission to come in. And a really important point here is that you need to make sure that you're applying and including in your application both the R61 and the R33 phases. If you don't have both included, you will be administratively withdrawn. So one of the advantages of this mechanism is with the phased approach, we have go, no-go milestones that determine whether or not a project will proceed from the R61 to the R33 phase. So this de-risks the project a little bit. And these need to be quantitative milestones that are proposed in the application. Reviewers will comment on them, as will we. And part of the advantage from the applicant perspective is that you can have, unlike in an R01 mechanism, you can have aims that are dependent on each other. And this helps de-risk that if you don't meet those milestones with the R61 phase, you won't continue on with the R33. So the entry criteria for this PAR The first one is there a strong biological rationale? So in the biomarker discovery space, there's a lot of overlap between the R01 space and this funding opportunity. And so if you're thinking about trying to identify a novel biomarker, one of the questions that reviewers are going to look at is whether or not there's sufficient biological support of the premise that this biomarker would be relevant for the context of use proposed. So you want to think carefully about how much evidence there is and if more is needed before you're ready to propose this as a candidate biomarker. And then, for this program, again, because we're looking at research tools and really think about these as product development grants, what's the unmet need in neurotherapeutic development or clinical practice? So how will the biomarker that you're proposing be used, and why would that be something that isn't already available, or how would it be an improvement over current standards? And then again, because this is a focus program, you want to make sure that your study design is set up to test the biomarker as a fit for purpose tool for one or more defined contexts of use. So within the research objectives of the PAR 22089, your R61 phase, again, is set up to do really proof of concept studies to test a hypothesis that a candidate biomarker or biomarker signature is an indicator of a particular concept. So that might be the biological process or the response to the intervention. And you need to make sure that you include clinical, that you're using, human samples, tissues, or data. And I'm highlighting the fact that animal studies are no longer allowed. And this is a change from the original PAR 19315. So please be aware of that change. And then you may also need to develop or optimize the detection method for how you're measuring the biomarker. And that would include preliminary analytical validation. So what are the minimal criteria you need to be able to measure your biomarker for the use you're proposing? And then the R33 phase is set up to do that initial preliminary clinical validation of the biomarker or biomarker signature for a specified context of use. And in this, development stage, this earlier stage, you might have, 2 or 3 contexts of use that you're proposing, but you need to be planning to test the biomarker's utility for each of those in your R33 phase, and thinking about what evidence is needed to demonstrate that this is ready to continue on for further optimization and development. So again, these funding opportunities, because they are really deliverable oriented, product oriented, the go, no go milestones are going to tell you what is that product need to show at the end of the R61 phase, and what do you need to propose in the application. And so examples here would be the minimal acceptable performance characteristics of the detection method, right? So what's the precision, accuracy, analytical sensitivity, dynamic range, and so on for the type of biomarker detection method that you're using. Can you get sufficient accurate results, recognizing that there will be further optimization, but what's the minimum you need to show that you can measure this biomarker adequately for that R33 proof of concept study. And then with minimal acceptable association of biomarker with the outcomes of interest. So here, what do you need to show that this biomarker is going to be useful for the proposed context of use, that it is associated with the outcomes of interest? What's the statistical association? So again, all these milestones need to be quantitative, and you really want to think about how your biomarker is going to be used and what performance characteristics need to be met to demonstrate that utility. So I'm going to now hand this over to Adisa, who is going to take us through the cooperative agreement funding opportunities. And Adisa, just let me know when you want to move the slides.
Thank you. Thank you, everyone, and thank you for joining as well. So just like we kind of stated at the beginning, we have 2 cooperative agreements mechanism, a U01 research project and a U44, which is a fast track only for small business innovations. So the U01 research project cooperative agreement lasts up to 5 years with no budget limit. But mind you that any applications that are requesting more than $500,000 is required to kind of go through an approval process at least 6 weeks before submitting the application. while the U44 SBRR mechanism is a fast track only mechanism, which is split up into two phases. The first phase, which helps in a sense focus on your feasibility, technical merit, commercial potential of the research project, can last up to 2 years, with the budget limitation, of course, of $700,000 per year. While the phase 2, which kind of helps you continue in the sense the work that was started in phase 1, can last up to 3 years with a budget limitation of $1.5 million per year. And an important aspect to note is that all budget requests must have an appropriate justification for the applicants. And if applicants are requesting, more than the limits, stated here, they would have to contact Neuroscience program staff to discuss that before submitting the application. Next slide, please. So an important aspect of these cooperative agreements mechanisms are the milestones. And for both the analytical validation, the clinical validation announcements, go/no-go milestones need to be defined, for each year, just like Carol stated in the prior announcement. And we do really emphasize that the milestones are clear, are quantitative, an emphasis on quantitative, that you specify in the sense of timeline for each milestones, that they have to be realistic and also definitive, because you go through an administrative review to transition from one year to the next. You should also, in a sense, provide, in a sense, sufficient details on the methods, experimental designs, and specify the quantitative criteria for measuring, in a sense, of success and the related rationale. Next slide, please. Thank you. So for the analytical validation of a clinical, biomarker, for both, the U44 and the U01, the entry criteria are the same. And in a sense, we do ask the applications, to be within the NINDS mission, right? Does it reduce the burden of neurological disorders? Is the project within, or does the project have a defined context of use? Is basically important that is clearly identified and also defined, and in a sense as well carefully considered, since it will be used to determine the experimental design and the level of analytical validation that is required. Finally, is there basically evidence of preliminary validation of the biomarker for the proposed context of use? Evidence should include preliminary estimate of the clinical sensitivity, and specificity and data that illustrates that the detection method has been developed and subjected to an initial evaluation of the precision as well as the sensitivity at a minimum. We do also, want product and each project should in a sense include any analytical validation, multiple testing site, which is really important. Next slide, please. So the research objectives should clearly define the outcomes of the project, and applications must propose to conduct, an analytical validation of the signature or the biomarker that has a well-defined proof of concept and biological rationale. You want to make sure that the application clearly describes how the proposed study plans to kind of optimize as well as standardize the detection methods. And those, and emphasis on rigorous, those basically have to be clearly defined and rigorously test the analytic and the pre-analytic variables to make sure that you have reliable and a broad and also reproducibility across basically multiple sites. You should also establish a protocol for the context of use proposed that could be used and also utilized across all sites to promote harmonization. Next slide, please. So there are two types of, next slide. Thank you. So there are two types of milestones that should be included when applying in the application. This includes performance milestones and also progress milestones. These milestones, again, must be clear, concise, and quantitative. We can only emphasize it so much. That obviously will be achieved during the period of the project. And the performance milestones could include, the description of the target thresholds for, sensitivity, specificity, accuracy, etc., of either, the biomarker or the biomarker signature, and also define, in a sense, an acceptable range, for the context of use proposed, and clearly demonstrate standardization and also the variation of coefficients across all sites. The progress milestones, on the other hand, can define or can be defined by either specifying, for example, the enrollment and the data collection goals each year, the contracts, the SOPs that will be utilized for each given year. And this could, in a sense, as well include, FDA consultation that needs to occur in the future. Next slide, thank you. So when moving forward and discussing basically the clinical validation, mechanism, the entry criteria remains the same as the analytical validation. But in addition to that, the proposed study has to, in a sense, have a detection method that has already been developed and optimized, which essentially acts basically, the detection method has undergone any analytical validation, i.e. basically what we kind of talked about in the previous mechanism, the initial evaluation of the sensitivity, specificity, precision, etc. And this basically, the level of experimental rigor, we can, put an emphasis on that again, that is basically necessary depends upon, the characteristic of, again, the biomarker, the detection method, and also the intended category of the biomarker, it being diagnostics or, monitoring biomarker safety, etc. That is obviously within the proposed context of use. Next slide. So the research objective, again, can mimic the analytical validation research objective that we spoke about, but it is important to note, two key points, for the clinical validation mechanism. So the research objective for the application should define and include, in a sense, what the biomarker acceptably identifies, measures, and as well predicts as a concept of interest within, again, the context of use. And secondly, incorporate a statistical threshold, emphasis on statistical, for the decision making, within basically the patient population, but most importantly, within a large and representative patient population. And especially when dealing with multi-site studies, designs have to basically ensure, that the biomarker validation is robust and is included and has an appropriate representative clinical popular representation, clinical popular patient population. Sorry about that. Next slide. Thank you. So as noted before, the performance and the progress milestones should also be included for the clinical validation mechanism. And again, not to be repetitive, they can mimic the ones stated in the analytical validation. But for these particular, in a sense, mechanism, you should establish how the appropriate cutoff/threshold, the positive and negative predictive values, will be determined. And in addition to that, obviously, the data quality assurance and control standards should also be demonstrated and standardized across all sites. And for, in a sense, the progress milestones, they can include, description of, the annual enrollment, et cetera, just like stated before. So now I'm going to pass it over to Carol to finish with.
All right, thanks, Adisa. So really quickly before we turn it over to Chris, just a couple of things to keep in mind. So non-responsive studies for all of these program announcements include ones where the therapeutic agents or devices as the primary purpose would be considered out of scope. Those are other funding opportunities. And then evaluation of clinical efficacy or effectiveness, right? Again, if the focus is on the therapeutic development rather than the biomarker, then they're out of scope. Natural history studies that don't include biomarker identification or validation as the primary purpose would be out of scope. With all of these, of course, many of these studies can utilize clinical trials, ongoing clinical trials, or data or samples from ongoing studies that might be useful for the biomarker qualification, but these program announcements aren't for those studies as the primary intent. And then again, animal models and studies using animal work are not in scope for any of these PARs now. And then you can be administratively withdrawn if you don't include some milestones in your application. And of course, anything that's outside of the mission of NINDS or NIA and NINDS for the case of PAR 22089. And these can be administratively withdrawn prior to a review. So you want to make sure that you think about this before applying. And then the very last thing I'm going to discuss here is just sort of a general reminder to everyone. You know, it's always good to read the funding opportunity announcement. There's a lot of information in there that we don't have time to go through in detail. You want to keep in mind that these are PARs, so they're special review criteria. These are reviewed at NINDS, and it's good as an applicant to both look at the instructions to you as the applicant, but also to the section of the reviewers that highlights what are the additional criteria specific to the funding opportunity, because that tells you a bit about what they're looking for when they're reviewing your application. And then again, you know, just to emphasize once more, the unmet need, how is this biomarker going to fill a gap? How would the detection method be an improvement? If that's the case, you know, where is the advantage here in the study that you're proposing? And then finally, study designs. You really want to make sure that the study design tests the biomarker signature usefulness within the context of use proposed. And because of that, you need to think about statistical analysis plans in great detail. They need to be described. We do have reviewers specifically asked to look at the statistical analysis plans for these applications. And because you're thinking about how you validate this as a tool and what those sources of variability are, you really want to make sure your statistician is involved in your study design right from the beginning. And I mentioned at the beginning that the FDA recently had a meeting about concepts and terminology for multi-component biomarkers, or what we're calling biomarker signatures. And this is discussed in the evidentiary criteria as well, but a couple of key points that you want to think about as you're proposing applications around a multi-component biomarker signature is, you know, what is the added value compared to a single component for a signature? What criteria will be used for including a feature or component into the final biomarker signature? So how do you decide to incorporate it? How will you evaluate that? And will the interpretation change when one or more of the components may drive the overall composite score or result? How will that drive your decision-making progress? Excuse me. All right, so with that, I'm really happy to turn it over to Chris Swanson-Fisher. So you are aware, NIH has a resource through the National Library of Medicine that they highlight the different data repositories and knowledge bases that are available and supported by NIH. And I'd encourage you to take a look at these and see what resources are available. And I'm highlighting here a few of the ones that NINDS is involved and supported of. And Chris is now going to talk about one of these, the Biomarker Biospecimen and Data Repository BioSend. Chris?
Thanks. Thanks, Carol. Good afternoon, everybody. Hi, as Carol mentioned, I'm Chris Swanson-Fisher, and I'm a program officer here at NINDS within the Division of Neuroscience. I primarily manage two of our NINDS repositories, BioSend, which I'm going to talk to you about in a minute, and also NHCDR, which is our NINDS Human Cell and Data Repository. But today, as mentioned, I would just want to provide an overview highlighting some of the capabilities for BioSend. But as Carol mentioned, there's a number of other NINDS research resources and broader NIH resources, which we certainly can refer you to, certainly to help support your broader biomarker research and fulfill the goals and needs. So, sorry, go back to the next slide. Thank you. Thanks, Rakanda. So what exactly is BioSend? So BioSend, essentially, it stands for the Biospecimen exchange for neurological disorders. This was coined by the current PI of the repository, Dr. Tatiana Farood. The repository is located at Indiana University. It's a U24 cooperative agreement. It's not a contract, but you can see on the right-hand side of the screen, there are just some screenshots I wanted to highlight for you that has some of the photos of the lab at Indiana University. If you go to the next slide. So as you can see on this slide, BioSend banks a broad spectrum of clinical biospecimen types, including whole blood, plasma, serum, DNA, RNA, CSF, saliva, urine, red blood cells. There's also a number of other biospecimen types as well. And we support banking of these biospecimen types from controls and a whole host of neurological disorders within the NINDS mission. We're banking Alzheimer's disease-related dementias. That includes vascular contributions to dementia. We have FTD, unfortunately, it's not located on here. We have dementia with Lewy bodies, also supporting biospecimen banking for Parkinson's disease, a number of Parkinsonisms such as CBD, PSP, and MSA. We also have a small closed study for chronic fatigue syndrome. We have a few cohorts for Huntington's disease, also spinal cerebellar ataxia, and traumatic brain injury. These samples are available to qualified researchers who may be interested in leveraging for their research proposals. So in order, we have many of them also are longitudinal, so it's just not cross-sectional collection, but longitudinal. There's over 7,000 subjects with biospecimens in the bank, and we probably have over 206,000 now biospecimens that have been received in the last 7 years to our repository, and a large host of them which have been distributed as well to qualified researchers around the world, both to academic and industry. So if you go to the next page, I'm going to speak a little bit more if you're interested on exactly how we can access those. If you go click ahead, yep, a few more times. Thanks. Just want to get to the first column there, and then there we go. There, that's fine. Thanks. So biospecimens from BioSend may be accessed through this online web request system, which are reviewed by what we call BRACs. Those are NINDS Biospecimen Resource Access Committees. They're disease-specific committees that are really comprised of extramural representatives from a number of cohort studies, those with expertise in clinical research, biomarker discovery and validation, and also biostatistics. So they're kind of like, think of it as small study sections per se. We want to really ensure that the samples are used appropriately within the NINDS mission. So we have an application that can be submitted through an online web form through our NINDS website. And it's received and reviewed on a rolling basis, approximately every 2 months. And so we've pretty much put forward a really straightforward process for the investigator determines what kind of samples they want, what cohort, and how many samples and what the volumes, and they determine that availability through working with our colleagues at the repository. They can submit an online application, which would include a small research strategy, about a 4-page limit, a biosketch for the primary investigator and any key personnel, and then also the table summary of the samples that are being requested. The BRAC would then convene, and then 2 weeks after that, if the samples are approved, we would put together a post-review process, if approved, to onboard with the repository and NINDS staff to discuss the timeline for distributing those samples. So right now we have BRACs for Parkinson's disease, also handling Lewy body dementia and Parkinsonisms. We also have for Huntington's disease, spinal cerebellar ataxia, chronic fatigue syndrome. We're putting together one for traumatic brain injury, which I'm very excited about. That should be coming live in the next month or so. And then we have a collaboration with NIA as well to review some of our FTD samples. If you go to the next slide. So yeah, That's perfect. So there's two primary ways that investigators can leverage BioSend capabilities for supporting your research, not just for biomarker research, for clinical research, for basic investigator-initiated studies as well. But I wanted to talk to you about this in the framework for your biomarker studies. So the first is banking. So if you're collecting biospecimens and that are part of your grant application, NINDS strongly would recommend that you leverage the BioSend infrastructure and collection protocols. I should note that the cost for that biospecimen collection must be included as part of your budget for your grant application. Now, this would include the costs of the collection kits, shipping those kits to and from the sites back to the repository, and some of the initial quality control procedures. We also expect that the IRB consent language, which will be associated with your study, should include language for sharing the biospecimens broadly with academic and industry. So we would suggest, and I'm happy to help you with this if interested, to email the BioSend staff, and you can work with their team, and their staff will help you prepare a quote. They have an online quote application request system, very straightforward, very timely, and happy to get that ball rolling for you if you're interested in banking with BioSend. If you go to the next, yep, you can go all the way through, Rakanda. Perfect, thanks. So the second is sample access. So as I just mentioned a moment ago, the samples can be accessed through an NINDS BRAC application. Now, you should note that we would request a letter of support from the BRAC to be included in your grant application, demonstrating approval. So plan accordingly. If you want to use some of these existing biospecimens at BioSend, I want you to put in a BRAC application first before you submit your grant application to Carol and Adisa, so that you can have that letter of support included with your grant application. That will help reviewers as well note that you've been approved to access those samples, and that may help you in your study section. So I'd also like to note that there's no cost to having those samples distributed to you, but you must demonstrate that funding has been put in place to run those assays as part of your research proposal. So the funding, you can apply through NIH, as we're talking about here. However, if you may also have co-funds from other funding sources, nonprofit organizations, institutional funding, et cetera, but we do want to require a letter that would be attesting and demonstrating these funds to be in place from a letter from your institution signing official. I've also, a couple more, Rakanda. Yep, that's what I just wanted to say. And then another more, there should be maybe can't read, but I just wanted to say my email address is down there as well. So if you have any questions about leveraging BioSend for your study or some of the other NINDS research resources, please do feel free to contact me. I'm happy to reach out to you and set up a call, discuss opportunities that we can work together to help support your research. So thank you. And I'll pass it back to Carol.
All right, thank you, Chris. And I apologize for my mouse that was a little extra sensitive.
No worries. Thanks.
All right, so I'm going to stop sharing and we're going to open this up to questions for anyone who has them. I see there's a few coming in now. And these slides, as Rakanda said, are going to go out after the webinar in a few days, and the links will be available so that you can look at those more. And of course, you're always welcome to reach out to us to discuss a project.
Okay, thanks, Carol. I'll go ahead and start reading the questions. Okay, our first question, did our submission in February, which include biomarker identified in both human and mouse model, is still okay? I assume the new policy only applies for the June cycle. Thanks.
Yes, that's correct. So the PAR 19315, any applications there still allowed animal work from the original discovery work. So those will be reviewed in June. And will be considered, just as the funding opportunity described.
Okay, second question. Does BioSend bank fibroblast cultures for deriving iPSC?
I can take that. Hi, Renee. Thank you so much for the question. That's a great question. And actually, what I would say is that our sister repository, that's NINDS Human Cell and Data Repository at Rutgers University, they handle collection and derivation and reprogramming for source cells, including fibroblasts, PBMCs, and then we also have a number of iPSC lines and edited lines as well for the research community. So if you have questions about fibroblasts, feel free to reach out to me and I'm happy to refer you to that repository.
Okay, looks like the next couple of questions are for you, Chris. Hi, Christine, great presentation. Is there a plan to include blood samples from hemophagic stroke and biopsy?
Yeah, hi, June. Thanks so much. Great question. So currently, we don't have hemorrhagic stroke in Biosyn, but it's certainly part of the NINDS mission. And I would be happy to work with you if you're interested in banking those. If you're interested in accessing samples from hemorrhagic stroke, feel free to send me an email and I can send you some resources that are ancillary to our efforts.
Okay, next question from Michael. Can Biosyn be used as a repository for other NINDS indications? Example, spinal cord injury?
Hi, Michael. Yes, absolutely. And so if you're interested, again, feel free to email me. Happy to work with you on setting up a quote and discussing supporting SCI efforts.
Okay, another one came in. I wanted to confirm that the SBIR deadline is June 22nd.
Yes, that's correct. So they have the same receipt and referral deadlines for both SBIR and academic. So we have a few more minutes. If anyone has any comments or questions they want to add, we'll keep it open for a couple minutes before we wrap things up. Again, I want to thank everybody for joining us. And thanks for joining. All right, I think if there are no other questions, we can.
Okay, here's one. Do we need to file patents for validated biomarkers? Any industry prospect?
It depends on what you want to do with it. So if you're planning on commercializing either the detection method or the biomarker itself, then you might want to do that, and you talk to your tech transfer officer. But it's not required as part of the program. Some applications propose open source tools, and it really depends on what you want to do and what your plans are. I don't know if Marianne or Ram want to comment as well.
Yeah, I think I would have had the same answer, Carol. And the other thing I just wanted to point out that it is important to at least, I think even in the new versions of these FOAs, you need to have at least address intellectual property. Is that right, Carol? There's still a clause in there?
Yes, that's right.
Okay. Yeah. And if not for no other reasons, just to show that you have permission to use the resources and reagents that you're using in the development of your biomarker.
Right, that's a good point. So I think the next question is for Chris.
Yes. Can NINDS biospecimens be accessed by for-profit companies?
Absolutely.
Okay. It's my understanding that genes as biomarkers cannot be patented according to a Supreme Court ruling in the US.
That is my understanding as well. But I will admit I am not up to date on the status of that. I don't know if anyone else on the
Not either, but I would say again, check with your tech transfer office when all else fails.
Agreed.
Any other questions? Getting some discussion here. We should have a open discussion time here.
Patent discussion, I think. Definitely.
We need a
We
That's
could have
workshop,
our tech
actually.
transfer
I
officer come
was
and
just
help.
thinking that we might do that as the next guest speaker for the next.
Yeah.
Excellent idea. Excellent.
All right. Well, thanks everybody again for joining. Thanks for all the panelists joining, and look forward to hearing from you. Any other questions as they come up as you're preparing your applications. And have a good weekend.